Hi, my name is Rachel Yee. I'm a junior at Washington State University. So my project is about these two genes called BD-WOX3 and BD-ligulus1, which are both in Brachypodium testacea, which is a grass model system. So we know what these genes, what these mutants look like in maize, and we're interested in what they look like in Brachypodium. So as you can see here, it's missing both the auricle and the ligule, which are these parts that you can see in maize. And without this part called the auricle, the leaf angle is a lot more upright. So as you can see here in Brachypodium, the leaf angle without it is a lot more upright. The main reason that we chose to study Brachypodium was that the genome is smaller, and since it is smaller, it's easier to create plants that correctly fluoresce the proteins that we're interested in. So with that, we were able to look at this through confocal imaging, and something we didn't expect to see is that the trichomes fluoresce and have protein accumulation. And then so we looked at the mutants to then kind of confirm if that was something that's actually true. And we noticed that in the mutants of this gene, the trichomes, which are these hairs here, were a lot more upright and they were fewer in number. So that was something we didn't expect to see, and that was definitely interesting. And then for BD-WOX3, we expect to see that they'll lose this part called the marginal domain, which is the outermost part of the leaves, and so that causes them to be a lot skinnier. And so when we looked at the mutants in Brachypodium, they did seem to be a lot skinnier, and something that we didn't expect is that they were also a lot shorter. So the blades were a lot shorter. In maize, they're a lot skinnier, but not shorter, so that was something we also didn't expect. And then when we looked at the histology of the Brachypodium, we noticed that there was this difference in this part called the wrapper and the tucker, which this is in maize. As you can see, the wrapper part, which is the outermost part, and the tucker part, it seems like they're different on either side. So the wrapper stays consistently on one side and the tucker on the other, but then when we look at Brachypodium, it seems like they switch every time. And that was something we didn't expect to see, and that causes us to kind of change how we think about this mechanism and how the lab is going to go forward investigating how the wrapper and tucker are initiated in those differences.
Well, you did a great job. I'm wondering, what are some of the methods that you learned over your REU program?
Yeah, I think one of the main methods that I learned was how to do dissections. I didn't have any skills in that beforehand, and it took me a lot of time to learn how to do them very precisely and accurately. So that was a lot of fun. And yeah.
Well, your poster and research looks great. Thank you.